So, our focus for this video is going to be on meiosis. Meiosis is a form of cell division. This type of cell division, when contrasted with mitosis, is all about making genetically unique cells. Mitosis is all about making genetically identical cells because we want those cells we make in mitosis to continue to carry out bodily functions. Meiosis, we don't care about bodily functions anymore. These cells are going to have a unique purpose, so they need to be genetically unique. These cells are going to go through fertilization. They're going to produce organisms for the next generation. The reason it is so important that these cells be genetically unique is that they are going to contribute genetic diversity to the population. Genetic diversity is the bread and butter of a species survival. They have to be genetically diverse in order to be a successful species. Genetic diversity is what allows a species to have different traits that will hopefully help them survive in adverse environmental conditions. So if the environment throws something really terrible at a species, hopefully they will have a genetic trait somewhere in their population that will allow them to be successful even if that happens. A little bit more info about meiosis. Meiosis is a process of cell division that occurs in the same part of the cell cycle that mitosis would. So these cells that start out that are going to go through meiosis will still go through G1, S, G2, but they're not going to go through mitosis. They're going to go through meiosis instead. And this picture here shows you what an actual cell going through meiosis looks like. So you can kind of see the differences between them. Only certain cells found in the reproductive organs can go through meiosis. Not every cell is going to do this. That's something that we see with mitosis. Theoretically, any cell could go through mitosis. Not so with meiosis. Only specific cells can go through meiosis. We're only going to replicate our DNA one time. We still only have one S phase. Even though we're dividing the cell twice so that we end up with 4 cells at the end, we only replicate our DNA one time. And what we're going to be dividing is homologous chromosomes. Now, in an earlier video where I talk about homologous chromosomes and I kind of explain chromosomal structure, I go more in depth into this. And if you're having trouble remembering what a homologous chromosome is, I would go watch that video. But homologous chromosomes, remember, are 2 versions of a chromosome. So humans have 23 different chromosomes. But in a cell in our body, we have 46 total because we have one version from each parent. We have a version of chromosome 1 that came from our mom and a version of chromosome 1 that came from our dad. When we go through DNA replication, we're making a copy of each of these versions. So at the end of replication, we get something that looks like this. We have version from our mom, copy 1 and 2, and we have version from our dad, copy 1 and 2. They're connected at the centromere. They have telomeres. They are still considered chromatids at this point. All of those things that we learned about chromosomes in the other video and when we talked about them briefly with mitosis still apply here. Nothing's changed about chromosomal structure. We're just making sure that you understand really clearly that what we're doing here is we have version chromosome 1 from our mom, version of chromosome 1 from our dad, and we've replicated them in order to get these X-shaped chromosomes you're familiar with. So meiosis goes through 2 stages. We call the stages meiosis 1 and meiosis 2 because we're super creative with names. So meiosis 1 is where we're going to start. We have prophase 1, metaphase 1, anaphase 1, and telophase 1. Those names should sound familiar because those are the names of the phases in mitosis. But now we have indicated that these are meiosis cell division stages because we indicate which meiosis phase we're going through, with meiosis 1 and meiosis 2 indicated after the name of the phase. So let's talk about prophase 1. Prophase 1 is where the nuclear membrane breaks down and centrioles begin to form and assemble the spindle fibers. The chromatin condenses into chromosomes, homologous chromosomes begin to pair up. All of these sound pretty similar to what we see in prophase in mitosis. But one big difference is going to occur here. In prophase 1, we're going to see crossing over occur. Crossing over occurs at the end of prophase 1, and it's a really important aspect of meiosis. Crossing over will only occur in prophase 1. There's no crossing over in meiosis 2. There is no crossing over in meiosis 2. So what does crossing over look like? Well, the first thing that we have to do is get our homologous chromosomes lined up together. So this is a process called synapsis. This is where the version of our chromosome from each parent find each other. So here we have our chromosome from dad, our chromosome from mom, and they're going to line up next to each other. Then crossing over is going to occur. That's where the telomeres start to entangle themselves. So they actually start to overlap the ends of these chromatids, those telomeres. And what they're actually going to do is switch sections of DNA. Actual genes are going to move from one chromosome to another. That is what crossing over is all about. It intrinsically or embeds genetic information differently in our offspring cells. We are making a completely unique strand of DNA here. We have guaranteed that although we have passed on our traits to our offspring, they're getting a unique set of DNA. This is very important for the survival of all organisms, making crossing over have that genetically unique sequence. So we have a quick video clip to show you so you can kind of see how this works in action. So here we have our chromosomes. They have lined up together. And they're going to walk you through how they actually go through crossing over. So here's the synapsis where they're lining up together. These are protein strands that hold the actual chromosomes together so that they're a little bit more stabilized. And you can see they're being pulled towards each other. Here's where the actual crossing over will occur. You can see they're lining up together. The chiasmata is the point on the chromosome where crossing over actually occurs. So, We are